Welcome back to the deep dive. Today, we are opening a file that on the surface looks like it's about agriculture, maybe a little bit about climate change.
Yeah, you see the headlines, you see
Yeah.
the cartoons of cows, and you think you know the story.
You really do. But once you actually crack the spine on the research, and specifically this incredibly dense, fascinating new paper by Zhang and colleagues from 2025, alongside some vital context from the Innovative Genomics Institute, you realize we're looking at something else entirely.
It is not just a story about farming. It's a metabolic thriller.
It really is. It's a heist story.
Right.
We're talking about a microscopic ecosystem where the main factory gets shut down, the waste products start piling up to toxic levels that should shut the whole system down. And just when you think the biological engine is going to stall, a silent operator steps out of the shadows.
And this operator doesn't just clean up the mess. It pulls off a metabolic coup. It steals the waste product and fuels a completely new engine that we didn't even know was parked in the garage.
That is a very dramatic but chemically accurate way to describe what happens in a cow's stomach. And that's our mission today. We are going to move way past the headlines. I think most people have heard seaweed stops cow burps.
Right. Feed them algae, save the planet. It's a great bumper sticker.
It is.
But the science behind why that works, and more importantly, what happens to the biology when you force that change, I mean, that's where the real story is.
Exactly. We aren't doing bumper stickers today. We are treating this hour like a graduate seminar. We are going to dismantle this paper figure by figure. We're going to talk about thermodynamics, partial pressures, gene expression, and structural biology.
We are going to get into the omics.
We are going deep into the omics. So for everyone listening, put your mental lab coats on. This is the advanced class. But before we get into the heavy biochemistry, let's set the stakes. Why are we spending an hour inside a cow's rumen?
Well, it comes down to the math of climate change. Methane is the villain here. According to the IGI data we have, methane is about 25 times more potent than carbon dioxide as a greenhouse gas.
And the Zhang paper cites an even higher number, right?
It does. They cite a global warming potential of 28 times greater over a 100-year period.
That's a massive multiplier. So a little bit of methane does a lot more damage,
Yeah.
Compared to the same amount of CO2.
A lot more. And livestock account for roughly 35% of all anthropogenic, that means human-caused methane emissions. That is a staggering slice of the pie.
35%. That's a third of the problem coming basically from barnyards.
Basically. And the issue is, unlike the energy sector where you can just, in theory, swap a coal plant for a solar farm or a wind turbine,
Right.
agriculture is a biological system. You can't just turn off a cow. The IGI describes it as a tough nut to crack because this methane isn't a bug in the system, it's a feature. It's how the cow works.
It's baked into the digestion. But before we get to the mechanics, we have to clear up the one misconception that plagues this topic. I feel like we have to do this every time cows come up.
The farts.
The farts. Every time this comes up in the news, it's cow farts.
It is not farts. The IGI transcript is very specific about this, and biologically it's crucial. It is enteric fermentation. It happens in the rumen, which is this huge fermentation vat at the front of the digestive tract. So strictly speaking, it is burps.
It's the exhaust coming out of the front.
Correct. The cow eats grass, the microbes break down that tough fiber, and the byproduct is hydrogen and carbon dioxide. Then methanogens turn that into methane, and the cow burps it out.
So the intervention we are looking at is red seaweed, specifically Asparagopsis armata.
Right. And this isn't brand new science. We've known for a few years that this specific seaweed is potent. The IGI source mentions studies where methane dropped by 98%.
98%, wow.
In this specific paper by Zhang, they saw a reduction of about 60%.
Which is still huge.
It's massive. If you could cut global livestock emissions by 60%, you are buying humanity a lot of time. The active ingredient is bromoform. It basically chemically sabotages the enzymes that make methane.
But here is where the deep dive really begins. Usually, we stop there, seaweed in, methane down, problem solved. We treat the cow like a black box.
Exactly. Inputs and outputs. But scientifically, if you just look at inputs and outputs, you run into a paradox.
A thermodynamic paradox.
Precisely. Because you have to ask, where does that energy go? If you block the exhaust pipe, the engine should choke.
Okay, let's unpack that analogy, the exhaust pipe. Because I think understanding the rumen as an engine is key to understanding this paper.
Sure. Think of the rumen as a big fermentation vat. It's an anaerobic environment, no oxygen. The microbes are breaking down feed cellulose, starch to create volatile fatty acids, or VFAs. We'll talk a lot about VFAs today.
And that's the fuel the cow actually runs on.
That is the actual fuel.
Right.
The cow doesn't really eat grass. The cow eats the VFAs that the bacteria make from the grass.
Okay, so the bacteria eat the lunch and the cow eats the leftovers.
In a way, yes. But that fermentation process releases hydrogen gas H2 as a byproduct.
Hydrogen is a waste product.
It is. And here is the thermodynamics part. Hydrogen is a potent inhibitor of fermentation. If the partial pressure of hydrogen gets too high in the rumen, the fermentation reactions stop. They become thermodynamically unfavorable.
So the chemical equation just won't go forward.
It won't.
It's like if you don't take the trash out of the kitchen, eventually you can't cook dinner anymore because there's no room.
That's a perfect analogy. And in a normal cow, the methanogens are the trash collectors. They are the hydrogen sink. They take that hydrogen, combine it with carbon, make methane, and burp it away. They keep the kitchen clean. They keep the partial pressure of hydrogen low so the other bacteria can keep fermenting.
So logically, if the seaweed kills the methanogens, the trash collectors, the hydrogen should pile up.
It should.
The kitchen should fill with trash, cooking should stop, the cow should stop eating.
The cow should get sick, digestion should halt, or at the very least, feed efficiency should tank because the fermentation is stalling out.
But in this study,
They didn't. In fact, feed efficiency went up,
they got more energy out of the food.
significantly more, up to a 74% increase in feed efficiency.
That is the mystery. The trash collectors were fired, the trash piled up, but the restaurant became more profitable. How?
That is what Zhang and his team set out to find. And the answer involves a microbe that, until recently, we barely knew existed.
I want to get to the hero, but let's look at how they set the stage. Because you can't just eyeball this, you need deep data. You can't just weigh the cow.
No, you need the omics. You need to look at the genetics of the ecosystem.
Okay, let's talk about the setup. Part 1. They used 12 lactating Holstein cows.
Standard dairy cows, yeah.
And they used a 3x3 Latin square design. Now, for the non-statisticians listening, is that just a fancy way of saying they shuffled the cows around?
It's a rigorous way of shuffling. In a Latin square, every cow cycles through every treatment phase. So cow A gets the seaweed, then the control diet, then maybe a different dose, and so on.
Why go through all that trouble? Why not just have a control group and a test group? Like, 6 cows on seaweed, 6 cows on grass.
Because cows are individuals. Just like you and I have different gut biomes. Maybe you digest spicy food better than I do. Or fiber cows very wildly. If you just split the herd, you might accidentally put all the good digesters in one group and completely skew your results. The Latin square lets each cow act as its own control. It removes the cow effect from the data.
Got it. That makes the data much cleaner. So they feed them this seaweed supplement. It was about 1% of their diet for 14 days, and they measure the output.
And the phenotypic results, the physical results, were stark. We mentioned the methane dropped. But look at the hydrogen.
I'm looking at table S1 right now. Hydrogen emission increased by 367%.
That is a staggering number. I mean, in any other context, that is a crisis. That is a hydrogen bomb going off in the metabolic pathways.
And yet the cows are fine. They're fat and happy.
To see why, they used metagenomics and metatranscriptomics.
We hear these terms a lot in modern biology. Let's define them so we are all on the same page.
Okay, so metagenomics is sequencing all the DNA in a sample. It tells you who is there. Think of it as the attendance sheet for a class. It lists everyone present in the room.
Okay. DNA is the roster. Steve is here, Sarah is here.
Exactly. Metatranscriptomics, on the other hand, sequences the RNA. RNA is the message the DNA sends out to build proteins. So RNA tells you what genes are being turned on. It tells you what the students are actually doing.
So metagenomics says Steve is in the classroom. Metatranscriptomics says Steve is currently writing an essay about hydrogen.
Perfect. Or Steve is currently sleeping. DNA won't tell you that. RNA will. If Steve is there but his genes are silent, he's not doing anything.
Now, there is a detail in Figure 1 regarding the database they used. They didn't just use the standard public libraries, right? This seems important.
This is a critical point for the validity of the study. Public databases for microbial genomes are still incomplete, especially for ruminants. It's not like the human gut where we've mapped almost everything.
Right.
If you rely only on public databases for a cow, you get a lot of unidentified species.
The dark matter of the gut.
Exactly. Just genetic noise that you can't assign to a specific bug. So Zhang's team built what they called a cohort-specific database. They took the samples from their cows, sequenced them, assembled the genomes from scratch, and then combined that with the public data. They call it RUMAG.
Rumen metagenome assembled genomes. Makes sense.
By doing this, they identified 3,119 bacterial species and 61 archaeal species. They mapped twice as many species as this standard tool, which is called Methane 4.
So if they hadn't built their own map, they might have missed the hero of our story completely.
Almost certainly. They would have seen the methane drop, but the mechanism would have remained a black box. They would have just seen unknown bacteria upregulated, and that would have been the end of it.
Okay, so they have the tools, they have the map. Let's look at the crash, part 2. The seaweed hits the system. What happens to the methanogens?
It's a massacre. But before the massacre, there was a case of mistaken identity in the control group.
Oh, what do you mean?
Well, look at figure 1b in the paper. This is the baseline, the normal rumen. For decades, the dogma has been that a bug called Methanobrevibacter is the king of the rumen. Usually people think it makes up 70 to 80% of the archaeal population.
That's the textbook answer. Methanobrevibacter smithii and its cousins. That's what I learned.
Right. But in this study, using shotgun metagenomics, they found Methanobrevibacter was a minority. The real dominant player was something called Methanomythilophilus. It made up about 66.7%.
So we had the wrong guy all this time. Why? Did these cows just have weird stomachs?
Probably not. It's likely a technology bias. We used to use something called 16S sequencing. It amplifies a specific marker gene to count things.
Okay.
Well, it turns out Methanobrevibacter has 3 copies of that marker gene in its DNA. Methanomythilophilus only has 1.
Ah, so Methanobrevibacter was shouting 3 times louder.
Exactly. The old tech was overcounting it because it had more megaphones. The new tech shotgun sequencing counts the actual organisms by looking at the whole genome. It's a great reminder that science is always limited by the tool you're holding.
So we have a new king, Methanomythilophilus. But then the seaweed arrives.
And the king falls. If you look at figure 2b and 2c, the archaeal populations, the methanogens, they just collapse. They drop from nearly 2% of the total microbiome to 0.02%.
That is essentially sterilization. It's almost gone.
It's a total collapse. And figure 2d shows the gene expression, the RNA, just flatlining. The lights went out.
So what specifically turned off? What were the key genes?
It's the entire assembly line for making methane. We see downregulation of FWDA, that's formylmethanefuran dehydrogenase is the first step, downregulation of MTRAH, the methyltransferase, and crucially downregulation of MPRAB.
MCRABG, I feel like we should highlight that one. That sounds important.
It's the most important one. That is methyl coenzyme M reductase. It is the enzyme that performs the final chemical step to create the methane molecule. The bromoform in the seaweed binds to the cofactor in that enzyme, a thing called F430, and just locks it up.
It puts a padlock on the ignition?
Effectively.
Yeah.
So the factory stops. And this brings us right back to the crisis, the hydrogen spike.
The trash is piling up. 367% increase in H2.
Right. Now, in a normal biological system, this is where things go terribly wrong. High hydrogen pressure stops the fiber-degrading bacteria from working. The Ruminococcus, the fibrobacter, they rely on low hydrogen to do their job.
So we should see a drop in digestion. The whole thing should grind to a halt.
We should, but we didn't. So the researchers started hunting for the hydrogen eaters. They looked at the hydrogenases, the enzymes that move hydrogen around. This is figure 3a.
Okay, and they split them into producers and consumers, right?
Yes.
Yeah.
The producers are the FAF types, iron hydrogenases. They kept pumping out hydrogen. The seaweed didn't stop the production. The bacteria that break down sugar were still making hydrogen.
So the leak was still there. The tap was still on.
Exactly. But then they looked at the consumers. The knife types,
Yeah.
nickel iron hydrogenases, and the type used by methanogens, that's knife 3C crashed. That's expected.
Because the methanogens are dead.
Right. But then they saw a spike, a massive upregulation in the knife 1 type.
Yeah.
This is a class of hydrogenase used by hydrogenotrophic bacteria, bacteria that eat hydrogen as food. It's off the charts. That's the smoking gun.
So someone is running a new hydrogen-eating operation, a shadow operation that just switched on.
And they trace those transcripts, the RNA messages, back to the source. And this is part 4, the hero.
So who is it? Who's our hero?
95.7% of those hydrogen-eating transcripts in the treated cows came from a single genome, a microbe called Duodenobacillus species, MGYG 0000293775.
MGYG. You know, that really rolls off the tongue.
It's a dark matter microbe. It hadn't been cultured before, it was just a sequence in a database. But in these treated cows, its abundance increased 58.3-fold.
58-fold. So it went from a nobody to the most important guy in the room.
It moved into the empty apartment left by the methanogens and threw a party. But the question is why? Why Duodenobacillus? There are other hydrogen eaters in the rumen. Why did this specific guy win the lottery?
This is the heist part. What tools did he have that the others didn't?
So they did a comparative genomic analysis. This is figure 3C. They lined up Duodenobacillus against 8 other potential competitors, and they found it had a unique metabolic arsenal.
Weapon number 1, what is it?
Weapon number 1, fumarate reductase, or QFR.
Okay, let's do some biochemistry. What is QFR and why does it matter?
So most bacteria in the rumen have an enzyme called SAQR. SAQR runs a reaction converting a molecule called succinate into another one called fumarate.
Okay, succinate to fumarate.
Duodenobacillus has QFR, which is designed to run that reaction the other way, from fumarate to succinate. And crucially, it uses hydrogen as the power source for this reaction.
So it takes fumarate, blasts it with hydrogen, and makes succinate, and in the process, it consumes the hydrogen.
Yes, that's the sink.
Correct.
But there is a supply chain problem.
Which is?
There isn't a lot of free fumarate just floating around in the rumen. It's a pretty scarce resource. So if you only had the factory, the QFR, but no raw materials, no fumarate, you still fail.
So where does it get the fumarate? It has to get it from somewhere.
That's weapon number 2, the DQ transporters.
The supply lines.
Right. Duodenobacillus has 8 different C4 dicarboxylate antiporters. These are basically little pumps on its cell surface. Compare that to its competitors who had far fewer.
And what do these pumps bring in?
It has a specific one, DCUA, that allows it to import aspartate and malate from the rumen environment.
Aspartate and malate. Are those common in the rumen?
Very common. They are amino acid derivatives and organic acids from the feed. So Duodenobacillus imports aspartate, uses internal enzymes like aspa and sumab to convert it into fumarate inside its own cell.
Wait, so it makes its own fumarate?
It manufactures the raw materials from stuff just lying around the rumen, then feeds it into its QFR engine, burns the hydrogen, and spits out succinate.
That is an incredibly elegant system. It's a complete vertically integrated assembly line. Aspartate, in fumarate, hydrogen added, succinate out.
And here is the kicker. Succinate is liquid gold for the cow.
Why? What does it do for the cow?
Succinate doesn't stay succinate for long. It is rapidly converted into propionate by other microbes in the gut. And propionate is the primary precursor for glucose in the cow's liver. It is the main energy source for milk production and weight gain.
So let me get this straight. The seaweed kills the methanogens. Hydrogen spikes. Duodenobacillus sees all this free hydrogen and says free energy. It sucks up aspartate, runs this internal factory, clears the hydrogen, and spits out a precursor to propionate for the cow.
That is the heist. It steals the role of the hydrogen sink from the methanogens. But instead of making methane, which is useless to the cow and bad for the planet, it makes propionate, which is food for the cow.
It turns a waste product into a fuel product.
Exactly. It's a metabolic rerouting and completely explains why the cows got fatter. The energy that used to be lost as burps is now being captured as propionate.
That is genius. It's a beautiful system. Now, you mentioned there was one more detail, a graduate level detail about the structure of the enzyme itself. This is figure 3D.
Yes. Figure 3D. This is for the structural biologists listening. It's a really neat piece of the puzzle.
I'm ready. Lay it on me.
They looked at the protein structure of that knife 1T hydrogenase, the engine eating the hydrogen. Usually these proteins are held together by something called an iron-sulfur cluster.
Like the structural beams of the protein? Or the wiring?
Think of them as the electrical wiring. They conduct the electrons to make the reaction happen. Most species have a 4F4S cluster. That's 4 irons, 4 sulfurs. It is held in place by 4 cysteines, which are a type of amino acid.
So 444, a very stable structure.
Right. But if you look at the sequence for Duodenobacillus MGYG 0000 is 293775, it has 6 cysteines.
2 extra. What does that do? What's the point?
It changes the geometry of the wiring. It allows for what's called a 4FE3S cluster. And in the scientific literature, this specific kind of weird structure is associated with oxygen tolerance.
Wait, isn't the rumen anaerobic? No oxygen allowed? I thought that was the whole point.
It is mostly anaerobic. But cows are big animals. They eat fast. They swallow air. Trace amounts of oxygen are always getting in. It's not a perfectly sealed system.
And oxygen is usually toxic to these enzymes?
It destroys standard hydrogenases. It oxidizes the cluster and breaks the circuit. But if Duodenobacillus has this armored, 4Fe3S cluster, it can keep working even when little pulses of oxygen get in. It gives it durability. It's more robust.
Oh, it has the supply chain, the transporters. It has the factory, the AQFR enzyme, and it has the armor plating, the cysteine cluster.
It was evolutionarily primed to win this competition. It was just sitting there waiting for the methanogens to get out of the way.
That is fascinating. It's like a sleeper agent that was perfectly designed for this exact scenario.
That's a great way to put it.
Okay, so Duodenobacillus saves the day. But you can't just delete 2% of an ecosystem without some fallout. Let's talk about part 6. What happened to the rest of the bugs? The ones that weren't the hero.
Right, because it's a whole community.
Mm-hmm.
And this is where we look at the C-enzymes. That's carbohydrate-active enzymes. These are the tools bacteria use to break down fiber.
And figure 4 shows a surprise here, right? Because I'm thinking feed efficiency went up, so they must be digesting more fiber.
A big one. You would expect fiber digestion genes to go up. More digestion equals more energy. That's the logical assumption.
But they went down?
Significantly. We saw a major downregulation in the Clostridia species. These are like major fiber degraders. Genes for breaking down cellulose, that's GH48, and hemicellulose dropped.
So the Clostridia, which are usually the heavy lifters for fiber, they stopped working.
They went on strike. Clostridia are very sensitive to hydrogen pressure. Even though Duodenobacillus was helping, the hydrogen pressure was still higher than normal. Clostridia couldn't handle the new work environment.
So if the main fiber eaters quit, how did the cow get energy? How did efficiency go up?
Functional redundancy is a key ecological concept. The Clostridia quit, but the Bacteroidota species kept working.
Ah, the scabs crossed the picket line.
In a manner of speaking, yes. The Bacteroidota appear to be more resistant to high hydrogen pressure. So the ecosystem shifted the workload. It fired the sensitive workers and relied on the sturdy ones to pick up the slack.
It's amazing how resilient this system is. It just finds a way. It adapts.
It does, and that shift changed the chemical output. We call this the VFA profile, the volatile fatty acids. That's in Figure 5.
We talked about propionate earlier, which is the big winner here.
Yes, but interestingly, we actually see a downregulation of the sus gene, which is succinyl-CoA synthetase.
Wait, I thought we were making more succinate, so why would a gene related to it go down?
We are, but this suggests something called feedback inhibition. The Duodenobacillus was making so much succinate that the system had to dial back the genes that regulate it. The tank was full, so it sent a signal to, slow down production elsewhere.
And what about butyrate? That's another key energy source for the cow.
This is really cool. The standard pathways for making butyrate were stable, but we saw an upregulation of what's called the lysine pathway. That's genes like COMBE and KDDD.
What does that mean in plain English?
It means the microbiome started fermenting amino acids, specifically lysine, to make butyrate. It was finding alternative routes to get the job done.
So the main highway is jammed. Okay, let's take the side streets.
That is the perfect metaphor. The seaweed blew up the methanogen bridge. The traffic, which is hydrogen, backed up. So Duodenobacillus opened a new tunnel, the succinate pathway, and the Bacteroidota paved a new road for fiber digestion. The result: traffic kept moving, and the city actually prospered.
So part 7, synthesis. What does this all mean for the future? We have this incredible paper from Zhang, and we have the vision from that IGI video. Let's connect the dots.
The metabolic model is clear now. Seaweed kills methanogens, hydrogen spikes, Duodenobacillus wakes up, imports aspartate, burns hydrogen, makes succinate, which becomes propionate, and the cow gets more energy. It's a win-win.
Right, but the IGI video brings up a really practical point. You can't necessarily feed seaweed to every cow on Earth every single day.
It's a logistical nightmare. Growing that much seaweed is hard. Getting it to range cattle cows that are out on a pasture and not eating from a trough every morning is almost impossible.
So what's the next step? Where do we go from here now that we know the mechanism?
The researchers, specifically Ermias Cabrera and Matthias Hess, who are featured in that video, are looking at the obvious next step. Now that we know the hero microbe is Duodenobacillus, can we just give the cows Duodenobacillus?
A probiotic, like a super yogurt for cows.
Exactly. Or, as the IGI video suggests, the pill. Imagine treating a calf just once. You introduce a gene-edited version of the microbiome, or you simply inoculate them with a high dose of Duodenobacillus early in life.
You establish the clean energy economy in their gut when they are babies.
That's it. If Duodenobacillus becomes the dominant hydrogen sink from day one, maybe you don't need the seaweed. Maybe the methanogens never get a foothold in the first place.
You inoculate the baby cow. Here is your super bacteria. And it stays with them for life. No methane, better feed efficiency for the farmer, and no daily supplements.
That is the holy grail. The video mentions using CRISPR to edit the microbiome directly. If we can engineer a rumen that inherently prefers the succinate pathway over the methane pathway, we solve a massive chunk of climate change. Remember, 35% of anthropogenic methane, without asking farmers to change their operations significantly.
That is the dream, a biological software update for the cow.
It is. But we have to be cautious. This study was 14 days.
Right. It's a short-term study.
We don't know if duodenobacillus stays dominant for a year or 5 years. We don't know if the methanogens eventually develop resistance to the bromoform, or if the duodenobacillus eventually exhausts the supply of aspartate in the rumen.
Nature finds a way, right? Maybe the methanogens find a workaround. There's always an arms race.
Exactly. Biology is an arms race. But what this paper proves, and this is the key takeaway, is that an alternative stable state exists.
That's the key phrase, alternative stable state.
We proved that a cow can function, and function better, without methanogenesis. That is a paradigm shift. We used to think methanogens were essential, like the liver or the heart. Now we know they are just one option. And frankly, they are the inefficient option.
They are the gas-guzzling option. Duodenobacillus is the hybrid engine.
I like that analogy. It fits perfectly.
So, wrapping up, we started with the cow burp problem. We established that methane is a heat-trapping monster. We looked at the seaweed solution. But the real discovery here isn't the seaweed. It's the bacteria behind the seaweed effect.
It is a discovery that the rumen has a backup generator,
Yeah.
a hidden system that was just waiting for the right conditions to switch on.
Which leads me to my final thought for the listeners. We tend to think of biology as fixed. Cows make methane, that's just what they do. But this study shows that biology is a marketplace. It's an economy.
Supply and demand.
Right. If you change the incentives, if you tax the methane, in this case with seaweed, and you subsidize the succinate by creating this high hydrogen environment, the market shifts. The economy reorganizes itself.
And nature abhors a vacuum. When we deleted the methane niche, something evolved, or was waiting in the shadows, to fill it.
It just makes you wonder, how many other silent microbes are waiting in the gut or the soil or the ocean? How many other duodenobacillus-type heroes are out there ready to save the planet if we just nudge the chemistry in the right direction?
That is the exciting part. We're just scratching the surface of the dark matter of the microbiome. We found this one because we looked. What else is down there waiting for us?
Well, on that hopeful note, keep looking for the heroes in the microscopic world. Thanks for listening to The Deep Dive.
Keep diving deep.
See you next time.